So here we're looking at a whole nephron. We have the glomerulus here, Bowman's capsule, and we were going to start talking about the proximal convoluted tubule. So you can see how it's all curvy and twisted, and so that's how you know it's convoluted. It's proximal because it's in close proximity to Bowman's capsule. So we know that if we have something proximal, we're going to go see eventually something that is distal. So proximal convoluted tubule is the region of the renal tubule just beyond the glomerular capsule, and its lumen is continuous with the capsular space. So the filtrate that entered the capsular space is now going to go into the proximal convoluted tubule. It's in the cortex of the kidney. Like I said, it's all twisted up. And if you looked at the wall microscopically, you'd see that it's made up of a single layer of columnar or maybe cuboidal cells. And the key thing about these cells is that they have microvilli. And microvilli, if I bring the notes, I have like a little picture up here. Microvilli, here's a little cell, and microvilli are just the folding of the plasma membrane. And what this folding does is it increases the surface area of the plasma membrane. So we're going to see that again when we are studying the digestive system and we look at absorption. So when you want more absorption, you want to have more of an area for secretion, you're going to have these microvilli. And also refer to what they look like under the microscope, which is a brush border. So what's kind of interesting then is you went to all this trouble to make the filtrate, and the majority of the filtrate is going to get reabsorbed here right away in the proximal convoluted tubule. Yeah. So the next part after the proximal convoluted tubule, so that's all the proximal convoluted tubule, the next part that you have is, if you can pull it away a little bit, is the loop of Henle.
Ooh.
Okay, is the loop of Henle. This here, and going up to here. So we're going to look at that. So two limbs to the loop of Henle. The descending limb, it's going from the cortex down deeper into the medulla, and then the ascending limb of the loop of Henle, which then leads into the last part that we'll look at, which is the distal convoluted tubule. So the descending limb, if you look at the wall of the descending limb, again, microscopically, you're going to see thin squamous epithelial cells in the thin segment of the loop. So the two loops can actually be divided into thin segments and thick segments, and we're not really going to go into too much detail about that. Same deal with the ascending limb. It passes out of the medulla and leads back up into the cortex, and you're going to again see thin and thick segments to that limb as well. So we'll talk maybe a little bit more about it, but really not go into too much detail about that but some of the textbooks do. So the last part that we're going to look at is the distal convoluted tubule. That's the last part of the tubule. And again, it's highly coiled or twisted. That's why it's called convoluted. It's just located here, and it's what's going to empty into the collecting tubule. It really doesn't have many microvilli, unlike the proximal convoluted tubule. So you're not going to see a lot of reabsorption occurring in the distal convoluted tubule. So that's it for the tubule. Then from the distal convoluted tubule, you're going to have emptying into this tubule right here. And this is referred to as the collecting tubule or also the collecting duct. And if you look here, you can see that there would be several nephrons, several distal convoluted tubules of different nephrons emptying into that single collecting tubule or collecting duct. And this collecting tubule or collecting duct is going to pass through the medulla of the renal pyramid. And you're going to have adjacent tubules joining together. And I'll show a different picture that shows that a little bit better.